1 second. We were discussing the diagram of the kidney, of the processes, I think we were discussing. Okay. Okay, here. Now look at this diagram. Okay. Now look at this. Sample 1 was taken from blood plasma that is coming from your renal artery. Okay, look at this. This bright red is your renal artery. This maroonish is your renal vein. Okay. Now the sample 1 is taken from blood plasma that is coming from your renal artery. Now it will have protein, it will have glucose, it will have urea, and it will have sodium ions concentration. Everything is present, and all of this is represented by each letter A, B, C, and D. Clear up till this point? I think yesterday Zoya, you were asking about where exactly what is being filtered. Okay, so this is a very summarized diagram as to at which point what is being filtered. Look at your sample 2. It passed through glomerulus and it entered your glomerular filtrate. Now look at this. Your flow rate is 100%. There is no protein. There was protein over here in sample number 1. There is no protein in sample number 2 in glomerular filtrate. Okay? Because protein molecules are too large to pass through that. Alright? Is this clear up till there? Okay. You have glucose as well, urea as well, and sodium ions as well. Why? Because they were small enough and they passed the glomerulus and entered your glomerular filtrate. Okay. Third sample is taken from your end of first coiled tubule, that is your PCT, proximal convoluted tubule. Okay, so just at the start of your loop of Henle, this is where your loop of Henle is starting, and this is where your loop of Henle. Okay, this was taken at just at your end of your PCT. From here till here is your PCT. This is your loop of Henle, descending arm or descending limb. This is the base, ascending limb of loop of Henle, and then you have your DCT from this part till this part. This is PCT. This is PCT. This is DCT. This is loop of Henle. And this is your collecting duct. Alright, now look at this. Flow rate has reduced from 100% to 20%. 80% of it has been reabsorbed when it's at the end of your PCT. There is no protein was already not there. Now there is no glucose as well. Now this is in a normal person. If it was in a diabetic person, then obviously you would have had some glucose concentration present. Urea is thrice, sodium ions are there. Okay, urea concentration has increased. It has become 3 times. That means all of the additional urea that was present in the blood not removed, that is also now added. Okay. Fourth sample taken from a collecting duct. Now look at the difference. Yes, sir.
This in the PCT there was a glucose, right? Now where did the glucose go?
It got reabsorbed back into the blood. It will not move forward with the filtrate. See, these red lines is all capillary surrounding it. Okay, so there is constant exchange of material.
Okay.
It got reabsorbed back into the blood. It will not be lost in the urine. Okay.
Okay, miss.
Now, some more reabsorption took place in entire loop of Henle in your DCT. And now look at your collecting duct. The flow rate has become 1%. 99% has been reabsorbed back into the blood. There is no protein that was not even there in the second sample. No glucose from third sample. Urea has increased 60 times and sodium ions have increased twice. Urea has increased 60 times and your sodium ions have increased twice. Is this clear? 99% of it has been reabsorbed back into the blood. Now is this clear? Yes, everyone, is this clear?
Clear.
Great. Now let's talk about homeostasis. Okay, it is the maintenance of constant internal environment despite the changes in the external environment. Despite the changes in the external environment. Even if it's negative 10 degrees Celsius, your body temperature will stay within the narrow limits. Okay. Now, a very good example is of controlling a blood glucose level. Okay, that is a very important part of your homeostasis. Why? Because cells need a constant supply of a steady supply of glucose when that allows them to respire. Right. Without this, they will not be able to respire. And if they're not respiring, they will not be able to release energy. Okay. Brain cells as well. Okay, they are also dependent on glucose for respiration, and they may basically die very quickly if they're deprived of it. Right. Brain cells constantly need to respire, and they will die if they're not receiving constant oxygen. So blood glucose is regulated by two hormones or controlled by two hormones. Concentration is controlled by hormones. That are released by the pancreas. If blood glucose concentration is high, insulin is released. Now, high means that above the set limits. Okay, insulin is produced or released. And if blood glucose concentration gets low, glucagon is released. All right. So you have insulin and glucagon. and both of them are released by the pancreas. Pancreas has a very, I can say unique feature that pancreas is both an exocrine and an endocrine organ. Endocrine, on the other hand, is a ductless gland. Okay, so it plays both roles. Pancreas basically makes your pancreatic juice. Okay, which flows through the pancreatic duct. Flows through the pancreatic duct into the duodenum. Alright. You have islets that are basically called islets of Langerhans. So they are a group of cells okay, that are scattered through the pancreas. They are scattered through the pancreas. Okay, which make up two hormones that are insulin and glucagon. But these are produced by your islets. Okay, main name is islets of Langerhans, but we just call them islets. Okay, so let's just call them islets easier. Okay, so they make two hormones. Your insulin and your glucagon. Two very important hormones. Okay. Now these hormones help the liver to control the amount of blood glucose because your excess glucose is stored in your liver in the form of glycogen. Right? And all of this is working on the phenomena of your negative feedback mechanism. All of this works on the phenomena of negative feedback mechanism. Alright. Basically 0.8 and 1.1 milligram per centimeter cube is the range of your blood glucose concentration. These are basically your set point. You don't need to know the values. Okay, I'm just giving you a value. Okay, this is basically a set point for blood glucose concentration. The point is, we need to understand when exactly this mechanism is triggered. Okay, that is what my point is. When exactly is this mechanism triggered? Pancreas and liver work together to keep the values within this range. Okay, if your blood glucose concentration gets too high, that means above this level, your insulin will be produced, like I told you. If the blood glucose concentration gets too low, that is below this level, then your hormone glucagon will be triggered. So in this negative feedback mechanism, there is a set point. Negative feedback means yes. See, negative feedback means the body works like in opposite direction to what exactly is happening in the body. For example, if your blood glucose concentration has increased, for example, you just had a meal, okay, and your blood glucose concentration increased, body will take measures to bring it down. It's working in the opposite direction, negative feedback. If your blood glucose concentration lowers, for example, you were fasting for the entire day, okay, and your blood glucose concentration has dropped, okay, so body will take measures to bring it up. Negative feedback. It's working in the opposite direction. Is this clear? Now, what exactly negative feedback is? Okay, great. So, like I said, that is a set point that is normal level that the system basically tries to maintain. Okay, and again there's a like set point like I told you that basically keeps it within your narrow range. Okay, if the level goes outside the range, that is maybe above the range or below the range, that will basically take trigger events or trigger some measures to happen that will bring the level back into line again. Okay, if for example blood glucose concentration increases, it will produce insulin to lower it down. Okay, and if it decreases, what will happen? It will take measures to bring it up and produce glucagon. Clear? Everyone clear? Any questions? Yes, Saad, Zoya, Ahmad, Umar, Ibrahim, all of you clear?
Clear.
Ibrahim, Saad.
Miss, what is the unit of the set point?
Unit of set point?
0.8. What does it measure? The number.
So you are asking me for the units of blood glucose concentration.
Yes.
Okay, it is. Let me write it here. It's in basically milligram per deciliter. There's another unit as well. I think for this value, I think it was something else, but let me check. There are different units, right? So mm, let me check the 0.8 and give me a second. Let me just Google it. If there are 2-3 units, let me check which values did I quote. I exactly don't remember the values right now. I just wrote them. I think it was just wrote it randomly. I think so. I'm not wrong. I don't think it's this low. Let me check 1 second. Homeostasis. Yeah, this is milligram per centimeter cube. Okay, yes. Deciliter, this will be very less. That's what I was thinking. This is milligram per centimeter cube. Okay, you also have it in millimole per liter. Okay. Or milligram per deciliter. Okay. You have 2-3 units for this. Right? Yes, Zoya.
Miss, how exactly is pancreas and endocrine and exocrine?
See, because it releases hormones both via duct as well and without duct as well. Now, in your body, there are certain organs that are only exocrine, that only has basically ducts. It only produces secretion via ducts. And there are some organs that have no ducts. It directly pours secretion into the blood. Pancreas acts as both. It also produces some secretions via duct. For example, this, pancreatic juice that flows through pancreatic duct. This is an example of exocrine. And this, glucagon and insulin are released without duct.
Okay.
Yes, rest of you clear?
Clear.
Okay, now let's talk about diabetes.
Clear.
Okay, so in type 1 diabetes, cells that secrete insulin die. Okay, and that is basically a response of your body's own immune system attacking it and destroying the cells in the pancreas that secrete insulin. Okay, in type 1 second, let's reshare it. Cells that secrete insulin die. Okay. As a result of body's own immune system. Immune system attacking and destroying the cells of the pancreas. Attacking and destroying the cells in the pancreas that secrete insulin. Okay. Symptoms are basically your dry mouth, blurred vision, feeling thirsty, heart rate, beating rate, all of them increase. Okay. So you have a lot of prominent symptoms. For treatment of type 1 diabetes, okay, what will you do? Control the amount of carbohydrate taken and take long-acting insulin. Control the intake of carbohydrate. Take long-acting insulin. And how will you check the levels of your blood glucose? Very simple. For testing or for checking, you can use a biosensor. Basically, it basically pricks your finger. Okay, takes the blood and tells the value of blood glucose. Okay. Second is you can use dipstick test. Dipstick is a test that you do it on urine. I mean, you don't do it, obviously, labs do it. Okay. And they dip the stick in your urine. Okay. And it changes certain colors. Okay. And if you have concentration of glucose in your blood, that means you're diabetic. Okay. Because apparently, according to what we have studied, there is zero concentration of your glucose in your urine. There is not supposed to be any percentage of concentration in your urine. Right? Because that means something wrong with your filtration. Okay. That means it means you're diabetic. All right. Clear? Clear?
Clear.
Ma'am, dipstick can
Yes.
come as a test in paper 6?
Maybe.
Okay, ma'am.
All of it. Okay. They can. But what will they ask you? They can take samples from 5 people. Okay. And then I don't remember if it has ever come, but it can. Okay. Ayla, why are you late? Yes. Meanwhile, rest of you, any questions? Okay. I will talk about controlling of body temperature. Very important. We have to see the structure of human skin. This is what your human skin looks like. Why is it so blur? Wait. I copy it again. Okay. I mean, it's understandable to an extent, right? Okay. So what do you have? Not zoom at that much. 3 layers: epidermis, dermis, and then your fat layer. This fat is basically the fat that is stored in your body, as subcutaneous fat under your skin. The excess fat that you consume. Okay. This sebaceous gland is basically producing sebum. Sorry. Okay. Then yes, Zoya.
Is this fat layer the adipose tissue?
Yes.
Okay.
Same. Okay, this produces sebum, that is basically your oil. Okay. And then I have your hair follicle. Okay. Basically your hair, okay, that you can see on your arms. Then blood vessels. Red ones obviously are oxygenated. It contains oxygenated blood. These are your arterioles, and your blue ones are your venules. This is your nerve ending, obviously that helps you feel pain. Sweat gland that produces sweat. Capillaries and melanocytes that produce melanin that gives your hair color. Color. In endodermis, your fat deposits. Okay, just like adipose tissue, it's also called subcutaneous fat. It's under the skin, subcutaneous fat. So sweat glands basically extract water and ions from blood and reduce sweat. Sweat glands extract water and ions from the blood and produce sweat. Okay. So this sweat travels up the sweat duct, okay, to the skin surface and is released to a pore, okay, obviously. And underneath all the structures, there is a layer of fat, like I told you. This fatty tissue is made up of cell that contains large drops of oil as well, sebum. Okay. This layer helps to reduce the body against heat loss and acts as an energy reserve as well. Okay, so this prevents heat loss. Prevents heat loss. Okay, and acts as energy. Then I have your hypothalamus. and it is at the center of your control mechanism that keeps internal temperature constant. It is at the center of the control mechanism okay, coordinates the activity of the body. Okay. It contains temperature receptors. Okay. That's temperature of the body via the blood running through. Okay. And if it is above 37 degrees Celsius, obviously your hypothalamus sends electrical impulses along the neurons to the parts of the body which have the function of regulating your body temperature. Again, it works with set limits. Okay, so there are temperature receptors. There are temperature receptors. Near the surface of the skin. Which sense the temperature of the air or water around it. All right. Now there are two things that happen. I mean, there are two conditions. One is when temperature rises, or other is when temperature falls. The changes that occur. Firstly, when temperature rises. Number one, hair lie flat. Okay, your erector muscles in the skin relax. So that the hair lie flat. on the skin. Okay, this allows heat to leave the skin through radiation into the air. This allows heat to leave the skin through radiation in the air. Second, I have vasodilation. But arterioles supplying the capillaries near the surface of the skin. Wider, that means dilated. Therefore, more blood flows through these capillaries. Near the surface. As there's a lot of blood near the surface of the skin, near the surface of the skin, heat is readily lost from the blood into the air. Third is when your sweat production increases. Sweat glands secrete more sweat. Secrete more sweat. Sweat lies on the surface of the hot skin. Of the hot skin. Okay, so the water in it evaporates, taking heat from the skin, thus cooling the body. So the water in it evaporates. And it evaporates, taking heat. Heat from the skin. Just all right. Thus cooling the body. When temperature falls, okay, muscles will contract in this. I think I have this written down. 1 second. One second, let me share the screen. One second. Let me just fix this. Look at this. Your hair will lie flat. Okay, yes, your erectile muscles in the skin will relax. This will allow you to sorry. Wait. I think I pressed the wrong one. It's supposed to be vasoconstriction. 1 second. Sorry, my bad. 1 second. I pressed the wrong one. It's supposed to be vaso. Sorry. 1 second. Copied the wrong one. I copied it from the top. Sorry. Okay, yes. Let me share the screen back. Okay. Now, in this case, your muscles will contract. Okay, when temperature falls, your muscles in some parts of the body contract and relax very quickly via a process that is called shivering. Okay, and they get energy from this via the process of respiration. Obviously, that provides them the ATP to do this, and in this, all the energy is released at heat. That's why and it's basically it's not intentional. Okay, it's subconscious movement. Okay, your body does it, and that basically releases heat, conserves heat. Okay, through the process of shivering. Heat generated in the muscles warms the blood as it flows through them. Blood distributes the heat all over the body. Then metabolism, okay, there's speeding of a chemical reaction in the other tissues that may increase, and this releases more heat. Okay, your hair stand up. Okay, The erector muscles in the skin contract, pulling the hair up on end. That means they stand up. Okay. If they will stand up, they will not let, they will trap the air layer. Okay. Obviously, we don't have that type of fur as, your mammals do, like polar bears and all, obviously. But at least, to an extent, you will see this, in winters that this happens. Obviously, it's not that visible in humans because, like I said, we don't have a layer of, warm blubber or warm thick fat, a thick layer of hair as well. We don't have that, right? But yes, we do have some hair. Okay. And that all, you can better see this in polar bears and all, that type of organism, but not in humans. But anyways, the main purpose is to trap your uh, layer of air. Okay, that basically helps you to preserve your except for preserve, maintain your temperature. Okay. Then you have vasoconstriction. So arterioles that supply the blood next to the surface of the skin becomes narrow, that means constricted, and very little blood can basically flow to it. Okay, like over here we had vasodilation, over here we have vasoconstriction. So very little blood can flow in the surface capillaries. So blood will flow deep under the skin, conserving heat loss on the surface of the skin through radiation, just like that happened in your uh, vasodilation. Okay, heat was being lost from the surface of the skin through radiation. Okay, sweat glands again, they will reduce the quantity of sweat that they produce, therefore no heat loss will occur. Okay, on the other hand, in your what was that? In when temperatures rise, okay, what will happen? There will be a lot of heat loss because sweat glands increase the production of sweat. All right, Claire, any questions, anyone? Yes, all of you. Zoya, Umar, Saad, Ibrahim, Ayla.
No question, ma'am.
Zoya, Ibrahim, Ayla,
No, miss.
all clear. Is this phenomena clear of when temperature falls and when temperature rises? What basically happens?
Clear, ma'am.
Okay, there was a good video on it as well, on excretion basically. I wanted to show you. Okay, 1 second. Let me just take it out. I'll just search it out, and then I'll share the screen with you. 1 second. Let me just find it. It's about excretion in humans. First, let's watch the homeostasis one because that is something we did right now, and then we'll go on the excretion one. 1 second. If it's about your feedback too, you can keep on explaining with the video as well. 1 second. Yeah, I think it's this one.
Did you know just sitting here right now, you're doing something absolutely remarkable? Well, your cells, tissues, organ systems, yes, we just leveled up those biological levels of organization, they're all working towards something called homeostasis. It's a state of balance. Yes, homeostasis means many things in your body. For example, that your blood stays within a certain pH level range. It means that your blood glucose remains within a certain range. It means your internal body temperature stays within a certain range. See, we've mentioned the major body systems before and that they work together. And today we're going to talk about how they work together using something called positive and negative feedback, and also how this relates to homeostasis. So many years ago, I had a pet bearded dragon. Her name was Debbie, and she was the best lizard ever. Debbie used to sit on our couch with me when I'd watch TV, and she loved to have her chin scratched. I even got her a bearded dragon leash so I could take her outside. Yeah, they make those. Anyway, Debbie loved her heat lamp. She would sit under the heat lamp on her rock, and when she got too hot, she would get off her rock and out of the heat lamp range and go somewhere else. She had a huge enclosure too because I wanted Debbie to be a happy lizard so she could find an ideal temperature. Why all this talk about Debbie? Well, Debbie is an example of an animal that some people refer to as cold-blooded, or a fancier term, ectotherm. We actually like the fancier term a bit better, though, because her blood isn't necessarily cold. Her body temperature can fluctuate with the environment. But not you. You are warm-blooded, or the fancier term, an endotherm. Your body works hard to keep the internal temperature it keeps. It's also a beautiful example of something called negative feedback. Before we define it, let us show you this example. Say you're in an environment that is very hot, like being outside in the Texas summer heat. That's typically hot. Thanks to nerves which can act as sensors, the brain notices this. It will send signals to counteract this variable. Sweat glands do what they do best, sweat. Heat is lost as that sweat evaporates off your skin. You may have some redness too. That's because your blood vessels are getting wider, dilating, in order to help get rid of that heat. And the result, whether you realize it or not, helps you lower your body temperature. But wait, what if you go inside now and the AC is blasting? To help you conserve the feedback is when some variable triggers a counteracting response in order to come back to some set point. If we consider that this whole thing is actually a negative feedback loop, we can see that the negative feedback brings the body back to the set point, which in this case is a stable temperature, keeping homeostasis. Negative feedback is also going on in the regulation of your glucose, your blood sugar. Okay, we're really simplifying this here, as we often do, but when glucose, blood sugar, is too high, one hormone that is released is insulin. I always imagine insulin as this hormone that makes the cells say, feed me, because it has the ability to make cells take in glucose, cause the liver to release glucose into the blood. There's more to the regulation of blood sugar than this, but you can see how this is negative feedback. You have counteracting responses here in order to keep homeostasis. So, what about positive feedback? Positive feedback is when, instead of getting a counteracting response to some variable, you instead intensify the variable. Positive feedback can be like more, instead of let's counteract this. The example that always stuck with me when I was a student is the example about the human baby being born. In biology classrooms everywhere, it's a classic example about how the baby is going to be born. More release of hormones will equal more actions help of negative feedback in maintaining homeostasis, and the role of positive feedback in many body processes. Well, we also need to understand feedback so we can understand what is happening when there is a problem in the feedback systems. One example, perhaps you've heard of type 1 diabetes. It's a disorder that can mean that your pancreas, which is an organ that is involved with making some hormones like insulin, is not working correctly. Insulin cannot be used in cellular respiration. The cells need to take the glucose in to make ATP energy in cellular respiration. Therefore, your cells need to be able to take in the glucose to survive. So many type 1 diabetics need to give themselves insulin and monitor their blood sugar because the negative feedback may not work as it should. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Or is this clear? Any questions? Alright, that's great. Then I'll keep it here for today. We're done with the chapter. Next class, inshallah, we're going to start with a new chapter. Alright? Alright, then I'll see you next class, inshallah.
Allah Hafiz.